We're going to switch gears a little bit with my presentation because we're going to dive a little bit into the molecular aspects of virus replication cycle. So it's going to be a little bit of a different talk. And what I'm going to do is I'm going to guide you, a little bit about superposition microscopy techniques, the virus replication cycle, and basically my small contribution to the field, with the goal of basically trying to convince everybody as much as possible how superposition microscopy really transformed the field of virology and has helped us to uncover secrets which we, or mechanisms that we didn't know existed before we were able to super resolve viruses. So what I'm going to do is, it's not working now, another fun. All right. Oh, yeah. Okay, now it's stuck. Okay, well, technical difficulties as always. Let's see. Oopsie. Let me try again. To focus now. It seems to be okay. Now it's working. Let's see if it detected the mouse again. Okay, now great. So yeah, what like I mentioned, I'm going to give you a brief overview about the superposition microscopy techniques and HIV replication, and then I'm going to go about several studies in which I looked at the HIV ENV distribution, HIV ENV mobility, as well as lipid ENV mobility on a fully infectious HIV assembly sites, and then a little bit of a preliminary data for my latest study where I started looking at the ENV distribution on vaccine candidate and founder strain viruses. So first, a little bit of an overview. So I think this slide appears into quite a lot of talks that fluorescence microscopy is is an amazing tool for scientific discovery. Seeing is believing. I mean, that's why we are here. So there are multiple advantages for why we're using a microscopes, fluorescence microscopy. Relatively simple and fast. Potential for live cell microscopy, high throughput. But then of course you have the often talked about limit to the resolution, and that simply is because we have the wave-like nature of the light, the interference, and the imperfect fluorescence light collection by the objectives. We basically are limited in standard confocal microscopy to around 250 nanometers in X-Y direction and around 600 nanometers in Z. So any object that is small, like a small, let's say like on this below here, 40 nanometer point object will be always appearing like 250 nanometers when viewed through a standard microscope. Well, but this has, of course, changed with the arrival of super-resolution microscopy techniques, and there has been quite a lot of them. And there is a lot of acronyms flying around right now. So actually, over here, most of the microscopes that are presented here by commercial companies have some form of a super-resolution nowadays. So the field has matured quite a lot, and you have different techniques, which basically, naturally, most of them rely on basically on-off switching of the fluorophores, as well as the precise knowledge of the location of the fluorophores. And they implement these principles in different ways to achieve basically the ability to super-resolve objects. So, for example, with HIV, you cannot see, this is ENV-labeled HIV, and you cannot see any details or other structures, and you get from this to this, where you can actually see a lot more than with a standard microscope. And so this is a current generation of the first generation of the super-resolution microscopy techniques, which were from around 2010, and now are very, very mature. But already there is a so-called, I guess, second generation of technologies already coming, MinFlux, for example, which offer even more resolution. So the field is progressing quite, is still progressing quite rapidly, but the techniques themselves are now very well established. So the one that I'm going to talk about is stimulated emission depletion microscopy, or STED microscopy. This is one of the techniques. And basically, on the top you see how the if you want to look at the virus that is labeled on the surface. If we try to excite it in the confocal microscope, the emission gives you diffraction limited volumes, and you really do not see anything in a final image. However, if we use the STED/resolve technique, which utilizes the depletion donut, which inhibits the fluorophores except in the center of 0 point, you can shape the PSF, point spread function of the microscope, to image fluorescence objects at much higher resolution. And in this case, classically, in XY at least, the STED microscopy oscillates between 20 to 40 nanometers in XY. Unless you use the 3D STED microscopy, then you still have the limitation in Z, but of course there is a 3D STED as well available. But the techniques that I'm talking about here mostly deal with basically just 2D STED. Now, a little bit about the HIV replication cycle. Just a bit of information about people who are not familiar, may not be familiar with the aspects of it. And so, yeah, HIV structure and proteins. So what is HIV? 140 nanometers envelope positive SS strand retrovirus. No need to, of course, mentioning how important of a pathogen it is. But from the molecular perspective and our replication cycle, I have to immediately highlight that the virus actually comes in an immature and a mature virus particles. So immature virus particles which just bud out are not infectious, and then the virus has to undergo maturation for it to be fully infectious. And this is basically highlighted here. First, we have the step of assembly and release of the virus. Afterwards, we have the maturation, in which the immature virus particle gets converted to the mature one. And finally, we have the entry, and then of course quite a lot of replication which is going on in the target cell. So here I also have a small animation which I did a few years ago, which I want to really highlight the dynamic aspects of the virus maturation, because this is a key component of the studies that I was involved in. So let's have a look in the what's happening in the supposed infected T cell. If we look really on a molecular level, so you have these molecules. These are GAG molecules which are floating around, and this is a single GAG molecule, which is a structural protein of the virus. And then basically they form dimers, they form multimers, and then this is RNA, which then at some point goes to the surface. This is not a fully scientifically accurate picture, but just to give the idea of what is happening when the new virus is generated. So initial aggregation of GAG then turns into self-aggregating system in which all the GAG molecules, the structural protein GAG, are going into this spot and form so-called immature GAG lattice. And this GAG lattice starts budding, basically creates a curvature, which then the virus will bud out. So at some point, enough GAGs form imperfect lattice, about 70% of the coverage, and then the virus buds out. And then there is a mechanism via escort machinery that the viruses then are able to undergo fission and then escape from the cell, bud out from the cell. Then afterwards, when that happens, the virus is free, but is in an immature form. So the GAG is not cleaved into its components. And basically, so what is happening there, concurrently with the release, or just after the release, you have the cleavage of the GAG into its components, capsid, matrix, and so on. And those then reassemble into the capsid, giving you the final shape of the mature virus particle. So this change inside, even on this cartoon, is dramatic. There is quite a lot of going on. So now a little bit to step, sort of put things into perspective. HIV really has benefited from the super-resolution microscopy. Of course, why? The viruses, like in the previous talk, the viruses are really small. So basically, HIV was one of the first pathogens that was put under the super-resolution microscope in early 2008, 2009, 2010s. So basically, the HIV is, I think, right now the still most studied pathogen by super-resolution microscopy. So people have targeted multiple parts of the virus replication cycle: assembly, release, virus architecture, entry, and post-entry stages. So really, all these studies, all of them have basically revealed previously unknown mechanisms of HIV assembly and entry. And then afterwards, I'm going to show you some of those in detail that I was involved in. But basically, I want to really stress that this reveals the enormous potential of these techniques for studying the virus replication cycle and virus cell interactions. Especially, I mean, not only HIV, but now SARS-CoV-2 and all the other important pathogens can benefit from basically all these techniques. So, I mean, I just highlighted some. This was studies. This picture was from all the review of mine. And since then, there has been quite a lot of other notable studies in which the image of the entire complete capsid was inside a nucleus, and new factors have been identified disrupting the virus surface and clusters, and so on and so forth. I mean, this is of course not an exhaustive list, just a few studies. But it's actually quite interesting that a lot of these groups, of course, this is a small same group of laboratories that publish this. However, now with all the microscopes that are available, I think a lot more people should be able to, a lot more groups which are not that familiar with these techniques should be able to get into these studies and utilize superposition microscopy. So now let's go to the first study that I was involved quite a long time ago, and that's basically looking at the HIV ENV distribution during the virus assembly and entry. And why is that? Back at the time, we didn't really know much about what's happening there. So this was actually the first study, one of the early studies to put a virus under the superposition microscope. So we knew that HIV particles incorporate much less ENV than theoretically possible. This is actually quite interesting that HIV particles, unlike even, let's say, influenza or other viruses, you have only between 7 or 10 ENV molecules on the surface. These are basically the ones that mediate the receptor interaction and virus fusion. We know that the ENV incorporation is influenced by the interactions with underlying GAG, and the ENV distribution may play a role in the virus entry because several ENV trimers are thought to be required for fusion. However, this has not been really visualized aside from some early electron microscopy studies. Also, ENV distribution itself may be influenced by interactions with GAG and thus affect entry competence of the virus. Because, like I said, here what's happening in the mature virus particles, the virus is not able to fuse with the target cell, and mature one can. So the aims of this early study were to basically visualize HIV ENV distribution using superposition microscopy, namely STED. To establish the effects of GAG processing or virus maturation on ENV distribution, and to observe the possible ENV rearrangements during the receptor interactions in wild-type and mutant particles. So here we basically utilize the system of where the virus itself is labeled with VPR GFP molecule. It's one of the accessory proteins of the virus. So we have the eGFP, which is in confocal mode, and then I labeled via FAB antibodies. I labeled the envelope protein. So on the left side, we can see this is how the signal, the orange signal is the important one. We see the diffraction limited spots. When we use the STED microscopy, we see suddenly that individual clusters of ENV on the surface of individual virus particles. And then basically what we did is we classified them and put them into several different groups: single cluster, 2 clusters, 3 or more clusters. And of course, this is not possible to use with the confocal microscope. So then we basically looked at mature versus immature virus particles. So what is the distribution there? And we found out that effectively when the virus is immature, we have a much more broad and multi-clustered ENV distribution than in a mature fully infectious virus particle, in which case the virus actually has predominantly only single clusters of ENV on the surface. So from the point when the virus matures, there is this reorganization of ENV molecules on each individual virus particle. Then we also looked at how the virus, how those ENV clusters colocalize with the CD4 receptors. So this is a 2-color STED microscopy in which an additional color is a blue, which is a CD4, and we saw that the individual clusters of ENV are oriented towards the CD4 and are interacting with it. And we also did additional mutations, including, I'm not going to go to too much details, where we looked at how does it interact with immature virus particles, or delta CT, or truncation of ENV C-terminal tail, which actually liberates it from interaction with GAG. And we found that immature virus particles are not able to cluster the ENV like before. But when immature delta CT mutation was put with the virus particle, we found out actually that the multi-cluster distribution of delta CT became a single clustered distribution similar to the mature virus particle, but only when you had the interaction with CD4. So actually CD4 interactions were able to recluster ENV into individual, basically to a single spot, which was very, very interesting because we basically with this we're concluding that HIV maturation of internal virus structures basically causes changes on virus surface. So this was actually, it's a novel inside-out signaling which hasn't been observed before. And also Another aspect of it is that ENV clustering on mature virus particles is important for efficient virus entry. Like I said before, the amount of virus HIV ENV on a surface per individual virus particle is very small, between 4 and 10 molecules. So by clustering them, the virus actually creates an environment in which it can actually fuse with this, fuse with the cell efficiently. So then the next step would be to look at the HIV ENV mobility, to extend the studies into the dynamic domain. And here, basically, like I said before, we observed the changes in distribution, but what are the mobility? Next, the question that was asked, we asked was, what is happening dynamically? Are there any mobility changes that are a potential cause for the ENV reorganization and maturation? So the aims for this particular study were to measure HIV ENV mobility on the surface of individual virus particles and to establish whether the maturation status alters this mobility rates. And to that, we turned into fluorescence coloration spectroscopy. And there has been already some excellent talks before by Torsten, by Don here, and Cecilia, who explained the principles. So I'm just going to go briefly and mention that, of course, fluorescence coloration spectroscopy allows you to extract information in form of an autocorrelation function. And from this, we can get average transit time of a molecule across the excitation spot. And of course, we can also get a number of molecules that are transitioning through that excitation spot. So with this average transit time, we can then derive a molecular mobility, diffusion coefficients. And here, of course, because we're dealing with viruses, we combine this with the STED microscopy, which allowed us, instead of the standard FCS, to look at the protein or lipid mobility on a subdiffraction scale. However, this was not enough. We needed to extend the concept of STED FCS a little bit further and to so-called scanning STED FCS. And this is because, like I said before, there is only very few ENV molecules on the surface. So you have about seven entities to label, and that is very, very limited amount of fluorophores that you can observe. And in FCS, if you point that excitation, and especially in the STED regime, then you bleach everything immediately. So we needed a bit more gentler approach. And in this case, basically, it's STED scanning, STED FCS, in which you sacrifice a bit of a speed of FCS acquisition, data acquisition, but you add a spatial component in which you can derive FCS data at the several different positions. This has an advantage of giving the fluorescence molecules time to relax a little bit and does not drive them into immediate photobleaching states. So we sacrifice a little bit of a speed, but we gain also a spatial component, which will be actually also important in the later studies. So utilizing scanning STED FCS, we looked at the virus particles. These are live particles, and this is a scan line in this case. Once again, VPR GFP particles, and then we collected in confocal the so-called carpets, in which this is x-axis and this is a time axis, and then we correlate across not the spatial component, but we across the temporal component and derive the autocorrelation function, and we can calculate the transit times. And here we came to a very, very big surprise, because we found out that the virus particles were mobility on ENV was incredibly slow. And this has been indicated already before that FCS may not be very reliable when we talk about slow molecules. So this was the case here. So those numbers has to be taken with a bit of a grain of salt, because I'm not sure if they represent the real diffusion coefficient. However, we were able to find that we had a consistent differences between immature and mature virus particles, when mature particles were slow, but immature particles were even slower, and that was consistent. And if we looked at the controls, we had no mobility, no correlated signal whatsoever in the fixed virus particles. So we wanted to actually see, is this just the ENV molecules that behave so bad, so slowly, or is it generally intrinsic mobility, intrinsic property of the HIV membrane? So in this case, we actually used a much more smaller molecule. In this case, just GPI anchor with a snap tag, and we labeled that first, and we looked at it in FCS in the cells. We got a pretty standard diffusion coefficient for this very small molecule, which does not, of course, trimerize. It has nowhere near the complexity of ENV. But when we looked at both mature and immature virus particles, we still found extremely slow mobility in this environment. So leading us to believe that the envelope is, as in the HIV envelope or plasma or the lipid envelope, is intrinsically immobile environment. So to conclude this, basically, we found that HIV exhibits a very low surface mobility, that is at least an order of magnitude a little slower than what you see on the cell membranes. We found that the virus maturation status does influence the mobility, and virus envelope is intrinsically very low mobility environment. And this is actually can be attributed to very high lipid order, extreme membrane curvature, and tight protein packing. And this is actually kind of you know, when we look at the cartoons of the virus particles, you know, all the cartoons, even the 3D ones, they all portray the viruses as if there is an empty space in it. So it gives it sort of a bit of a false reality of what actually the virus looks like. So if you look at the molecular simulations or how virus looks like on the inside, you can see there is an absolutely packed environment with all the different proteins, all the different, or of course, genetic material. And so combined with the tight curvature, no wonder it's such a low mobility environment. Even more so, much more so than a cell, which of course also has a, you know, on the previous visualizations you could see on how packed it is in cells as well. But this is even worse. Then no, the mobility did not increase. Exactly, with a CT mutant, exactly, which is much more free to move, we found out that also was around 0.02. So once again, intrinsic, it's about the membrane rather than actual, you know, whatever GAG ENV interactions you have. So then once again, extending this a little bit more, we wanted to see how well this works in actually fully infectious viruses. And we looked actually at the lipid and ENV mobility at HIV assembly sites. And why is this important? Because, well, first of all, a bit of a background. HIV membrane is highly enriched in sphingomyelin and cholesterol. And the previous consensus was that the virus assembly and binding may occur at the pre-existing lipid raft domains, or, you know, liquid order domains, whatever you call them, but it's a pre-designed environment, so to speak. But we wanted to test a hypothesis instead to check if, instead of pre-existing domains, does the assembling virus particles, does it create its own unique lipid environment on the virus assembly site? So to this end, we have basically determined the lipid and protein mobility inside and outside of HIV assembly sites. And like before, we use the scanning STED FCS, but here we combine it with a fully infectious virus model. So here's the cells that have been infected, Jurkat cells, post-adherence. 20 minutes afterwards, you have appearance of the assembly sites, which we then imaged using this time elliptical scanning STED FCS. This actually gives a bit more consistent sampling speed across all the positions, and it gives us the advantage of being able to sample both exactly inside the virus assembly site and just right outside of the virus assembly site. So we can look at the molecular dynamics inside and outside of the points of interest. So in this quite dense slide, we basically look at the several different molecules. We look at the PIP2, which is very important for the binding of MA to the plasma membrane, cholesterol, sphingomyelin, phosphatidylethylamine. And we found out that at least for lipids, in a lipid study, only PIP2, but not cholesterol or SM or PE, has actually highly reduced, cholesterol as well. Cholesterol and PIP2, but not SM or PE, have a highly reduced mobility at the basically budding early assembly sites. Then we extended this study to also confirm that this is the same for ENV molecules as well. So in a nutshell, we found out that we believe that the budding, this indicates that during the budding, HIV creates its own specific lipid environment right when it's just during the early budding sites. And it's doing so by selectively restricting lipids at the virus assembly. This environment combined with immature GAG lattice interactions leads to ENV trapping and its incorporation into the nascent virus particles. So I am going to also now finish off a little bit with just some preliminary studies of ENV distribution, revisiting the ENV distribution that I did almost 12 years ago, but this time to look at the vaccine candidate and the founder strains. So All previous work has been carried out with the so-called lab-adapted strains of HIV. And those, whether infectious or not infectious, they're still model systems which may not reflect the reality, which are the viruses that circulate in patients. And founded strains derived from patients may have different end characteristics. And also, this is important for vaccine candidate particles, because obviously, if you're designing the vaccine that is based on a virus particle, HIV vaccine, it is important that they should mimic the end distribution or the virus, bona fide virus particle as much as possible. So here, the aim was quite simple, to image end distribution and later mobility. I don't have the mobility data of virus candidate particles. And then later down the line, we would like to compare the results with the founded strains of HIV. So I have here just a little bit preliminary data of the patient-derived N with from using the VLPs with a patient-derived N with broadly neutralizing response. And this is a wild-type N. And what we immediately notice is how little ENV is on the surface. And that is actually attributed to GP120 shedding. So in this regime, the virus vaccine candidate is not a very good one because you have an immediate lot of shedding. And you actually need additional mutations, C-terminal truncation or mutation that binds the basically GP120 and GP140 domains of ENV together to retain more ENV on the surface. However, even then, and I still haven't quantified all the data that much, but even then we're already seeing that the virus particles, vaccine candidates seem to have a multi-clustered ENV distribution rather than the single cluster that you see in the mature virus particles. So this is with collaborator in Madrid. We are going to then look at the virus particles which mimic complexity of the real virus a little bit more. So just also a small sort of on a technical note, I am, after revisiting this particular study, after doing this same, I was amazed how easy now it is to take those images. 10, 12 years ago when I was taking these images with a STED microscopy, it took quite a lot of time to align the microscope to basically even get the images. Whereas right now with a modern STED microscope, and I can imagine probably with the other techniques as well, it took me really 30 seconds. With a click of the button of the auto-align microscope, bam, I have the images. So right now the technology is so much more mature and so much easier. You don't have to worry about alignments. You don't have to worry about all the problems that we had with the early microscopes. You can just get to the data. So hence, really, the take-home message that I want to drive is that superposition microscopy is a very powerful tool for studying HIV. And it does have a potential to make high-impact discoveries. But with the availability of the equipment right now, these techniques can now easily be utilized by a larger number of research groups for studies, of course, not only HIV, but all other pathogens. And I would like to just also end with the little bit of advertisement, is that if anyone is interested in superposition microscopy, we have a platform in Barcelona. I know there is a very good platform in Montpellier right now, but I know it's very, very busy. So just a few hours away, we'll have a STED microscope. We'll have several microscopes in the high-level biosafety. So if you're interested in doing some research, please contact me. And with that, I am going to finish. And these studies have been done over the years, so over several groups which I was in. This is, in Heidelberg, Hans-Georg Kreuzlich and Barbara's group. Then Christian's group in the Oxford. And with Cyril and Delphine, we did amazing collaboration as well. And right now, the studies that I do is with my affiliated group at IRSI Caixa in Barcelona. And with that, I will thank you very much, and I'll take questions.